So thank you very much for joining us, and we'll go to the first presentation, which is from Dr. Alicia Estevez from IRTA in Spain, who's going to talk about meager.
Okay. I'm not good at presenting things as Dinos, but anyway, I will do my best. Well, my presentation is just about what we have done with meager during all these years. And first thing is just to show why, well, Dinos already explained why we selected this species. Is because the market in the European Union tried to change from a small or slow-growing fish like sea bass and sea bream that has, they are sold between 350 and 600 grams to bigger fish that can be processed. So we selected these species that Dinos already presented. And among them, meager is one of them. Meager is considered as a good candidate because it's widespread along the Mediterranean and also in the Atlantic coast. Being Senegal, the Bay of Dakar, the southern part, the southern limit of the species. It has the advantage that it has this species that reaches up to 2 meters in length and 50 kilos in weight. It's important commercially and has a global distribution of fast growth and an excellent flesh quality. Because of this flesh quality and high yield, they can be or have innovative added value. No? So there have been a lot of efforts in the European Union and in the European fish farmers to develop these species. There are another species around the world that has already been cultured, like the Argyrosomus japonicus in Australia and Japan, or Silus gilberti or Stiaena umbra around the North American and South American countries. The product is very attractive because it has lean fish, very low fat in the muscle, high quality marketable products, reaches a relatively high commercial size very rapidly, and has a low conversion rate between 0.9 and 1.2. Started the culture of meager in Europe started in the 1997, produced in captivity in France from, I think it was from wild broodstock. They started selling, the first juveniles were sold from France, and then spread all over Europe. Now in 2017, the production has reached almost 8,000 tons, with Egypt producing 16,000 tons, but it's not clear what species they are producing. There are hatcheries in Greece, Spain, and France, and it's being grown in different companies all around Europe, mostly, well, the Mediterranean. The price, at least in Spain, is between 13 and 14 euros per kilo, and can be processed not only with big fish, but only with small size fish, 600 grams to 1 kilo. As Dinos already said when he presented the project, the first thing we did with this species was to ask researchers and producers which were the bottlenecks for each species. And in the case of meager aquaculture, there was not a really bottleneck in reproduction, although there was a need to fix a protocol for for hormone induction and management of the spawnings. There was a need of studying the genetic background of the broodstock that are spread all over Europe, and select several genetic tools for genetic breeding programs. In the case of larvae, there was not bottlenecks, but there was some bottlenecks already identified, such as the high degree of cannibalism in these species, and the problems with you and the on-growing, because the species usually distributed in the bottom of the sea cages, and the fish companies don't know how to manage the feeding of the on-growing fish. In the case of fish health, there were several diseases were identified, such as granulomatosis, chronic ulcerative dermatopathy, and the monogeny and parasitism. So what DIVERSIFY promised at the beginning? In the case of reproduction and larval husbandry, develop spawning induction methods, characterize genetically all the broodstock in Europe, and improve and develop new genetic tools. In the case of larval husbandry, develop appropriate weaning protocols because the larval rearing was quite well established. What we did was, the first thing was characterize genetically all the available broodstocks in Europe. They were 432 meager breeders were sampled from 13 different centers from 7 countries, studying the 18 microsatellite markers. And what we found was that there were 3 populations of different broodstocks in the Mediterranean. 2 that were quite close, and another one, the one in Turkey, that was quite separated. So it was quite different. The second thing we did was to identify genetic tools, and markers, and the description of the transcriptome, especially to future genetic breeding programs. So identify at least the genetic linkage map and several markers and SNPs. Okay. The next thing we do was establish a protocol for pair crossings and using induction, hormonal induction. And what we got was that we can have up to 17 consecutive weeks of spawnings after induction of 1 male, 1 female. And the spawnings were every 2-3 days after the hormonal induction. So it is reliable to establish genetic breeding tools using this technique, this induction method. We also established a good protocol for managing the eggs and sperm, establishing a good protocol of what to do and how much sperm used to have a good fertilization of the eggs. So it is written in the slide. But anyway, if you want to know every protocol that we have established, it is already in the manual that we have provided and is uploaded in the web page. In the case of the larval rearing, we established, I haven't said, but all the work done with the reproduction was done by the HCMR and IRTA. In the case of larval rearing and weaning, it was done in IRTA. And we did several experiments with trying to establish a good weaning protocol, or at least advance the weaning of this larvae that usually occurs at day 20, advance it to day 12 or 15. We found big problems with cannibalism among the larvae, but it can be done. But if several measures to avoid this cannibalism are put in place, like reducing the light, increasing the number of doses, and try to isolate the big fish that eat their siblings. In the case of nutrition and health, I'm trying to explain in different parts what we did. In case of nutrition and health, in nutrition there were already quite a lot of work already done, but still there were some nutritional requirements of the larvae and juveniles that were not well established, especially what refers to vitamin requirements. So we did several experiments with larvae and juveniles, establishing the requirements of several vitamins. In the case of health, there were work done to establish the immunity and of the ontogeny of the immune system in the fish, in order to later on establish a good protocol for vaccination and develop vaccines, of course. Also, we studied the problem of systemic granulomatosis and the chronic ulcerative dermatopathy. What we obtained after working hard. First, this work was done with the University of Las Palmas de Gran Canarias, and they highlighted the larval weaning diets, especially in terms of requirements of vitamin C, E, A, D, and K. And what they found is that, for example, in the case of vitamin D and A, it seems that this species is very sensitive to hypervitaminosis. And in the case of Menadion, it also has some problem of toxical effects. In juveniles, they also tried to work mostly with the requirements of fatty acids in the juveniles, especially in terms of N3 PUFA. And what they found was that if you reduce the quantity of N3 PUFA in the diets for on-growing fish, they have problems with steatosis in the liver. And if you increase the levels of this N3 PUFA, this steatosis is not produced. They tried to establish the effects between vitamins and antioxidant vitamins and essential fatty acids in the incidence of granulomas, but they couldn't find really an explanation about the granulomas. In the case of health, this work was mostly done by the HCMR, and later I think Vantelis will explain also the work they have done in health. One of the problems, very big problems in meager during the on-growing and especially in CKs, is the presence of granulomatosis. Granulomatosis, there are some granules that you can find everywhere in the body of the fish, mostly in the intestine and the liver, but also in the heart or even in the eye and the muscle. And it gives a bad aspect, a bad I don't know how to explain it, but in order to the consumer perception, a bad image of the fish. The problem of granulomatosis is supposedly due to the no-cardiosis, but there was no problem caused by no-cardiosis. It is considered that it's not a problem caused by no-cardiosis. There are several other nutrient origin of the problems of granuloma. They thought that perhaps vitamin D can decrease, but there was no effect. The content of phosphorus also in the diet, it seems that they have improve the condition of the fish. Also, they tried with different vitamins without any results. So it is not clear why granulomas is caused. It's supposedly due to some nutritional problem, but it's not really, we don't know really what is the nutrient that is involved in this disease. In the case of chronic ulcerative dermatopathy, it is well known that it's due to the quality of the water that you use. If you use borehole water, the problem appears. But when you move the fish from this borehole water to marine water or CKS, the problem disappears very slowly, but disappears. So it's good to have this in mind, that if you are going to produce the juveniles using borehole water, you have to move the fish as soon as possible to seawater to avoid this disease. I haven't explained, but also in the ontogeny of the immune system, we work with the IRTA, with the University of Aberdeen. But the information is still on working and publications, so I haven't brought any more information about that. Okay, and also because Dinos also insisted in trying to make a shorter presentation. The third part of the things that we were promised to work was about grow-out husbandry, especially as I told you before, the behavior of these fish in the CKs. Try to know how to improve the feeding methods to get advantages in the rearing. And also, of course, the development consumer market analysis and see how we can sell these products to the consumers and attract the consumers to buy these fish. In the grow out, we work well in the beginning using the first producers found that there were groups of fish that grow very very fast and others that grew very very slowly. So what we thought it was that perhaps putting the fish, the slow growers, separated from the high growers, could have a compensatory growth, as we find in sea bream or sea bass. But in this case, in meager, this compensatory growth doesn't exist. The slow growers always grow very slowly, and the fast growers always grow very fast. It is good in one hand to establish genetic breeding programs, but in the other hand, is bad because the slow growers usually have a 6 months delay in the grow, and of course they have a clear economical impact in the producers. So it is good to know which are the slow growers and the fast growers, and select the fast growers, of course. We also studied the effect of the light and the distribution of the fish in the sea cages. It was done in the ECMR by Nikos. And what we found is that usually during the day, the fish distribute in the bottom of the sea cages, and during the night they go up to the surface. So we thought that perhaps different lighting conditions will improve the feeding of the animals. And the experiment was done in the experimental sea cages in Greece. One was covered by a shadow, and the other was with natural light. But they couldn't find any difference in growth. So it seems that this was not a good, or the shading of the sea cages didn't improve so much the growth. Okay. We also tried to establish what were the feeding habits of these species. So we did 2 different experiments. One was done in IRTA using auto-demand feeders and program feeders to see how they behave during the feeding. And we thought that perhaps the auto-demand feeders would work better with these species, but no, we couldn't find any difference between both auto-demand and program feeders. They grew exactly the same and had the same conversion rate. In Greece, they work from another point of view, trying to use the automatic feeders, the normal ones that they use in the sea cages, but try to put some attracting methods to the fish, to attract the fish to feed when the feeders were working. So they used 2 things to attract the fish to the feeders. One was to use air bubbles as an attractive stimulus, and one was using light. Both were quite good for the fish. They learn very well and were well attracted by these 2 stimulus. But again, there were no differences when they tried this in the sea cages. And also they tried with a different feeding system. Instead of distributing the feed only in the surface, they use an S-form submerged feeding system that worked quite well. In the case of market, the work was done with IRTA and also the Wageningen University, I think. What they did was to produce 3 different products, fillets divided in double portions, a salad with fish, and also a fish burger with the shape of the fish. And they did a customer study, and with information and without information. So saying which species they were eating and which species, and saying or not, giving that information or not. And the acceptance was quite good. They tried the salad and the fish burger, and the products were well attracted and well considered by the consumers. So this is what we did in the project. Finally, we did a workshop in Barcelona last month with several producers and researchers, and we provide a technical manual for meager where we put all the information that we have done, we have collected during all these years for research. So the manual is uploaded in the web page, and you can have access to it, and you will have all the information that I have explained today. Finally, we found that it's important to continue doing research in these species, especially to reduce the problem of cannibalism. I think that is shared with some other species that we have been working in this project. Also continue working with the sea cage on growing, especially helping or guiding the producers how to improve the feeding in the sea cages. Because when we try to explain them how to do it, well, they have their own methods, and like, for example, keeping the fish 1 week in starvation. That is crazy. But anyway, I think that we need to teach them or to help them how to improve the way to feed the fish in the sea cages. And finally, to try to solve the problem of granulomatosis, because it's a real big problem, we still don't know which is the cause of this disease. So, and this is all. Thank you.
Thank you very much, Alicia. Okay, you have any questions for Alicia? Huh?
I have gone too fast.
Well, that's okay. It's always better to have time than to run out of time. The issue of differential growth with the slow growing, we did look into some markers for that, right?
Yes, and I know that Costas is working on that. Yeah, we collected some samples, and it seems that there is some marker. Yeah.
Okay. Because recently I was at a farm in Cyprus, and they showed me fish that they were 1-year-old fish that were 1 kilo, and 1-year-old fish from the same batch that they
4
were
kilos.
150, 200 grams.
Oh no, really?
Yeah.
Okay.
And they say that they see it all the time.
Yeah.
So it's
Yeah, there are fast growers and slow growers, and there is no compensatory growth. If you separate the slow growers, they don't grow better. No? Yeah.
okay.
We'll see with Costas.
Yes, Patrick.
Alicia, I see that the problem of cannibalism, that is not unique in meager, we have the same with pike perch, and to some extent is always creating big bias when we are trying to determine the nutritional requirements or etc. Because some larvae are simply feeding on the conspecifics.
Yeah. Yes.
Do you have some ideas how to manage this issue, to improve it?
In cod, in Norway, they have been working quite a long time with that, with the problem of cannibalism, and they have found that the domestication of the broodstock has a good part of that. So if you are working with larvae from wild broodstock, they are more cannibalistic than if you are working with an F2 or an F3 already born broodstock. So it seems that domestication of the broodstock has a very big effect. But so we would like to try that, to see if the larvae produced from wild fish and the larvae produced by an F2 or an F3 broodstock have differences in cannibalism. Probably they will.
Okay. They explain how it works. It's just because you are selecting the less aggressive fish, and it is some kind of inheritance of this behavior?
Yes.
But I think, Alicia, perhaps there is also a possibility to use the phenotypic variability existing in a wild population, because I think that according to different strains, there is a huge difference for these critters, and perhaps we have interest to select initial good population for that point. But
the problem is that usually we go to the fast growers fish, and perhaps fast growers are the more cannibalistic. So I think that we have to check what we select. Yeah.
Just a comment. I know that there is a big issue with the cannibalism and the size variability. But another way, apart from the genetic selection, as you said, to handle it is the husbandry practice that you apply during larva rearing.
Yes.
And we know very well that if we apply semi-intensive methods or, let's say, more natural methods, then we don't face this problem because we allow all the fish to grow in a better way, and then we just need to handle them and make a sorting at the proper size, and then you overcome the problem.
We know, but most of the fish farmers, most of the hatchery don't work in mesocosm. They work in intensive methods.
But they can work always with a lower density, which is much closer to mesocosm.
No, what they do, because I was talking with them during the workshop with meager, what they do is to start grading the fish at day 20. They start grading the fish, big fish and small fish, at day 20, really. Yeah. So there were some questions there.
Just a second, Alistair.
Yes.
Thank you very much. Alistair Lane, European Aquaculture Society. I wasn't able to get to the workshop in Barcelona, but I have two questions. One is, has the work that you've done over these years now been able to, have you been able to contribute to the ASC standards on meager, which are lumped with sea bass and sea bream, for example? You said that there are differences there, and I understand those differences. And then the second question is, in production at the moment, what would be stocking densities that are used, and how might that have an impact on the fast and slow growers?
Yeah, well, I don't know about the EAS, yeah. Which I don't know about
the
that.
Aquaculture Stewardship Council.
Don't know really.
Okay.
In the densities during the larval rearing, for example, we usually use 50 larvae per liter. And during the on-growing, I really don't know, but how many kilos? Around 20, 25? 15.
15.
Okay. Yeah. So 15 kilos per cubic meter. Okay.
Mr. Meisert.
Yeah, thank you very much. I just had a comment, and that was regarding the many kind of losers you have, or small fish you get, and that's the phenomenon we're seeing with many marine fish. It's a big diversity of size after hatching, and I think we are not good enough with feeding them. So we had the same, for instance, with turbot years ago. Yeah. We already had to grade away maybe 10-20% of the small turbot because it didn't perform.
Yeah.
And we've seen the same with cobia. We see it here. If you look to pike perch, you see the same enormous diversity. And I think then selection and would solve the problem in a couple of generations. Yeah,
yeah, we know. Yeah, the problem is that with meager, they are still not genetic selection methods. They are not established yet. It's too early for this species. So we need to know what kind of genetic selection we do. If it will be for fast growers or slow growers, or high cannibalism or low cannibalism. We have to check that.
I think you should go for the fastest growers. They will eat whatever they find, so just give them enough feed and then you solve the problem.
Not that much with meager. Yeah.
I wonder if there's any, if we know, not from published results, but from talking with farmers, whether the fish that coming from Le Poisson, who are the first ones that have started a breeding program, if their behavior, this size differentiation is less, it is, Nico? It's not less. So the breeding has not solved it yet, or the behavior.
Apparently we have not yet found the proper genes that we need to select.
Yeah.
We need to study a little more to find what to select on.
Yes.
It's not only the growth. I mean, the fast growers does not solve our problem. The fast growers just repeat the problem.
You want some?
I think it's a general problem in aquaculture because we have already selected fish according to growth of flesh quality, but we need to promote to find population which represent a trade-off between different functions and to use several traits.
Yeah.
According to growth, of course, but also behavior, also sensitivity to stress and things like that.
Not all the new species and not all the species that we have in aquaculture has so cannibalistic rate. Meager is very high.
Yes, I know.
It starts very early and continues up to the juveniles, because sometimes with the juveniles we have survivals of 1%. It's 1-2%. It was very, very, very low.
Yes, Alicia, but I think.
And once the cannibalism is established, you cannot avoid it.
Yes,
it's crazy.
10 years ago with Patrick, we have worked, for example, of different populations of perch coming from different ecosystems, and we have seen clearly that there is some populations that are very more aggressive than others.
Yeah. Okay. Yeah, but in case of meager, is too high, really.
Okay. Anybody else has a question or a comment? Okay, thank you very much, Alicia.